Good afternoon, children. Today, let us discuss the chapter Biomolecules. Biomolecules. Biomolecules, children, listen carefully, use your time. When though the classes are going on through online, utilize your time, do the exercise. Any doubts are there through WhatsApp, you can clarify. So here the chapter name Biomolecules we are discussing. This is an important chapter relevant for this NEET examination point of view as well as for both theory and NEET. It is important one. So biomolecules means what are the biomolecules? The carbon containing molecules. Biomolecules means the carbon containing molecules present in the living organisms. Those can be called as the biomolecules. Biomolecules means what are the biomolecules? Most appropriate definition for biomolecule, the carbon containing molecules. Carbon containing molecules. Carbon containing molecules present in living things. Present in living things are called as biomolecules. Called as biomolecules. The carbon containing molecules present in living things, those can be called as the biomolecules. Suppose if you want to analyze the biomolecules, our body, plant or any animal, what biomolecules are there? How can we analyze the biomolecules? One method is there. What is the method we are using for analysis of biomolecules here? Here there is a trichloroacetic acid method. To analyze the biomolecules in living tissue, we are using a method. That method is trichloroacetic acid method. You see that analysis of biomolecules in living tissue. Analysis of biomolecules. Analysis of biomolecules in living tissue. How can we analyze the biomolecules in living tissue? There is a method. What is the method here? Trichloroacetic acid method. You see, this is a trichloroacetic acid. Cl3COOH. How it will be called here? This is a trichloroacetic acid. It is called as trichloroacetic acid. By using trichloroacetic acid, we can analyze the biomolecule. For analysis, what we have to take in, we can take, suppose, analysis of biomolecules from plants. If analysis, otherwise animals. Suppose it is a plant or this is an animal. What we are taking here, mortar and pestle. One mortar, this is a mortar has been taken. Into this mortar, what we have taken from plants, we will collect vegetables. What we will take here, vegetables. These vegetables, whatever, tomato, lady's finger, brinjal, any vegetable, these vegetables have been taken, vegetables. Here, this is a mortar. We are taken into mortar and pestle. There is a pestle is there. It is by using pestle and mortar. Into the mortar, vegetables have been taken. And into this mortar, what we are adding? Chemical. What is this chemical? Trichloroacetic acid. By adding trichloroacetic acid, we are grinding. Into this mortar, vegetables have been taken. By using pestle and mortar, vegetables have been grinded by adding the trichloroacetic acid. Then afterwards, what the mixture will come? This is what we are doing here by using muslin cloth or cheese cloth, we can filter by using muslin cloth. What we are using, muslin cloth or cheese cloth, what we are doing here, we can filter. We can filter. So while we are filtering, what happens here? Some whatever the quantity it has been filtered, that is called as the filter rate here. That is a filter rate. What the material it has been filtrated, that can be called filtrate, which would not be filtrate, that which will be called here this filtrate fraction or filtered fraction. Filtered fraction of which will be called filtrate. Not filtered fraction of which will be called not filtered fraction, retinate. This is called retinate. Simply, what you can understand filtrate means here, suppose you have to take like this here. Here, this is a, what is here? One container. On this container, what we are keeping? One cloth. Then we are pouring this here, grinding mixture. Some mixture is coming into the container. What is coming into the container? Which will be called filtrate. That filtrate is also which will be called acid soluble fraction, which can be filtered into the container, that is called acid soluble fraction. Some here, some content, it would not be filtered, which is retained outside the cloth. That which will be called here acid insoluble fraction. This is which will be called acid insoluble fraction. Another name of acid insoluble fraction, retinate. So what you can understand from the plants, we will collect vegetable. Vegetables have been grinded by using mortar and pestle. Then afterwards, the extracted material, it has been filtered. Here, this is a container. On container, we can keep a cloth, we can filter. Some component, some fraction has been comes down, some fraction retains outside the cloth. Which will retains outside the cloth, that can be called retinate, which is coming down filtrate. The retinate is also called acid insoluble fraction. This filtrate, it can be called acid soluble fraction. So that the biomolecule, acid soluble molecule, acid insoluble molecule, two types. If it is for plants, but animals, same thing is applicable for animals. But instead of vegetable, in case of animal, what tissue has been taken? Liver tissue has been taken. Here, for this analysis of biomolecule, in the given plant or animal tissue, we are using trichloroacetic acid method. But in this method, if you want to estimate biomolecule in case of animal tissues, we have to take liver tissue. If we want to analyze biomolecule in plant tissue, we can use the vegetables. But by using mortar, pestle, either vegetable or liver, they have been grinded and filtered. Finally, what conclusion has come? In this analysis here, in acid soluble fraction, what molecules are coming? In acid insoluble fraction, what biomolecules will be there? Then we have to see this acid soluble biomolecule, acid insoluble biomolecules. These biomolecules, they have been, how these biomolecules, they have been classified here. These biomolecules, acid soluble fractions. Here, acid soluble fraction biomolecules. Acid soluble fraction biomolecules. Then acid insoluble fraction. Acid insoluble fraction biomolecules. Acid insoluble fraction biomolecules. But acid soluble fraction, which biomolecules are present in acid soluble fraction? There is a amino acids. What would be present here? Amino acids are present in acid soluble fraction. Next one, what would be present inorganic ions? Inorganic ions. Inorganic ions means calcium, magnesium, ions will be present here. Sodium, calcium, magnesium, like these minerals can be present in acid soluble fraction. Acid insoluble fraction, what biomolecules can be present here? Nucleic acids. What would be present here? Nucleic acids will be there. Next one, polysaccharides will be there. Here, polysaccharides are there. Nucleic acids, polysaccharides. Then the most thing, what is this here? Lipids. Next one, proteins. Here, these are the 3, 4 molecules. Here, nucleic acids, polysaccharide, lipids, proteins, these are there, acid insoluble. But these acid insoluble fraction molecules, how they can be called here? They can be called as the here biomacromolecules. Normally, they can be called biomacromolecules. Why they can be called as biomacromolecules? These acid insoluble fraction, protein, lipid, polysaccharide, nucleic acid, these are insoluble fraction. These insoluble fraction molecule, how it will be called here? Macromolecules. Why they can be called as a macromolecule? Those size is more than 10,000 Daltons. Here, what is the size here? More than 10,000 Daltons weight will be there. So they have a more than 10,000 Daltons weight. Therefore, here macromolecule. But here, what would be there here? There is up to this year 800 Daltons. These micromolecules, what is the size here? 18 to 800 Daltons size it will be there. Here, these biomicromolecules, acid soluble fraction molecule, 18 to 800 Daltons weight will be there in acid soluble fraction. Here, so that these acid soluble molecules, they can be called as the microbiomolecule. These are the macrobiomolecule because those weight is what would be there more than 10,000. 10,000 or more than 10,000 Daltons will be there. That can be called as the macromolecules. Now you see lipid is there. What is the lipid here? 800 Daltons here. The lipids are 800 Daltons only. But why they can be occurs in insoluble fraction? Lipids, they would not be solubilized. In acid here, this lipid, it is not solubilizing. While we are grinding leaves here, actually lipid is fraction of plasma membrane. While we are grinding, this lipid composition would not be crushed. Therefore, it would be occurs in the remains of the plants only. Henceforth, here lipids, where it is not coming into filtrate. What is the biomolecule? Occurs in insoluble fraction. Answer lipid. Lipid is a small molecule, not a macromolecule. But here, even though it is a small biomolecule, but where it is present here, along with macromolecule, it occurs in insoluble fraction. Why? Lipids, they would not be crushed properly. Therefore, it would not be, here comes into the filtrate. So it is staying in insoluble fraction. So what are the micromolecules staying in insoluble fraction? Lipids. Like these nucleic acid, polysaccharide, proteins, lipids, and inorganic ions, amino acids. These are the different biomolecules occurs in the cell. Some according to solubility, acid soluble fraction, acid insoluble fraction. In acid soluble fraction, what will comes here? Micromolecules, 18 to 800 Daltons biomolecule occurs in here acid soluble fraction. Acid insoluble fraction, which have more than 10,000 or 10,000 Daltons weight would be there. Those are the macromolecule. What will comes under macromolecule? Nucleic acid, polysaccharide, protein, lipid. These comes under this here acid insoluble fraction. Normally, insoluble biomolecules are macro. Only exception for lipids. Lipid is having 800 Daltons here weight, but it is staying along with insoluble contents. And now let us discuss here what are the cell composition. Different molecule cell composition. Let us see the estimated cell composition. various biomolecules composition. Estimated various biomolecules composition. Cell. Estimated various molecules composition in the cell. If you see this in the cell, this is the different molecules composition. Molecules and concerned molecules. You see this here, water. How much percentage of water is there? 70 to 90%. Total in the cell, how much percentage of water would be there? 70 to 90%. Next here you see proteins. How much percentage of proteins will be there? 10 to 15%. Protein composition in the cell, 15%. Now you see nucleic acids. How much percentage of nucleic acids will be there? Nucleic acids, 5 to 7%. Nucleic acid composition, 5 to 7%. Next you see what is this carbohydrates. How much percentage of carbohydrates are there? Carbohydrates are 3%. Carbohydrates will be there. Next here lipids. How much percentage of lipids are there in the cell? 2%. The estimated composition of cell lipids are 2%. Then what is the composition of ions here? Less than 1%. It will be there. So different compositions you see here water. The huge proportion of the cell is occupied by water. Why? Because water is medium for various metabolic activities. This is protein synthesis, lipid synthesis, vitamin synthesis, respiration, excretion, whatever the metabolism is taking place in the cell, it has been done in the presence of water only. Therefore, here, what would be their highest proportion has been occupied by water. It is what is the stake here? 70 to 90% of cell is occupied by the water. Proteins, how much percentage of proteins will be there? There will be a 10 to 15% protein composition is there. Then here nucleic acid. How much percentage of nucleic acids will be there? There is 5 to 7% nucleic acid composition is there. And carbohydrates. How much percentage of carbohydrates will be there? 3% carbohydrates are there. Then lipids. How much percentage of lipids will be there? 2% lipids will be there. Then here ions. How much percentage of ions will be there? Less than 1% ions are there. Next one, these biomolecules, they have been classified on the basis of involvement of this growth and development here. These can be considered as the primary metabolites, secondary metabolites. According to size or solubility, acid soluble biomolecule, acid insoluble biomolecule. But same biomolecule, how they can be classified here? Primary metabolites, secondary metabolites, as per their involvement in the metabolism. So here another classification of biomolecule, we can see primary metabolites and secondary metabolites. On the basis of involvement of biomolecules. On the basis of biomolecules involvement in growth and development. Biomolecules divided into primary metabolites, These biomolecules classification. How these biomolecules classified here? Primary metabolites. Primary metabolites and secondary metabolites. Secondary metabolites will be there. On the basis of involvement of biomolecules in the metabolic activities, they can be considered as primary metabolites, secondary metabolites. That means the biomolecules are metabolites which are directly involving in metabolism. They directly involve in metabolism. Those can be called as the primary metabolites which are directly involving in the metabolism, those can be called as primary metabolites. Example for these primary metabolites here, amino acids. Amino acids are directly involving in the air metabolism. Alcohols, here these are also directly involving in the metabolism. And now you see whatever these metabolites here, indirectly involving in metabolism or indirectly helping for growth and development. Metabolism means, suppose in place of metabolism, indirectly involving in growth and development means same only. Because metabolism responsible for growth and development. Therefore, here what would be there? Directly involving in growth and development or metabolism, such metabolites can be called primary metabolites. Normally, primary metabolites are there in animals. Primary and secondary metabolites are there in plants. Secondary metabolites where they can be formed plants. But secondary metabolites, what purpose using secondary metabolites here? Welfare of man. In plants, primary metabolites are there, secondary metabolites are there. Secondary metabolites here, they have been produced in plant but not used by plants here. Indirectly they are helping, but here what purpose? Plants, they are not using the secondary metabolites. Those products taken outside for welfare of man, here for economical aspects of man, secondary metabolites are using. So what are those secondary metabolites? They have been used in different sectors here. Let us see this here, secondary metabolite charts here. The secondary metabolites are secondary metabolites. Secondary metabolites. Secondary metabolites are, what are those here? Pigments. These are the pigments, alkalides, terpenoids, essential oils, pigments, alkalides, terpenes, taxins, lectins, drugs. Here, these are the different secondary metabolites. Pigments, what are the examples for pigments? Carotenoids. Keratinide, not chlorophyll. Keratinide is a secondary metabolite. Next to anthocyanin. Anthocyanin. Alkalides, morphine. Opium poppy plant, morphine is there. Codeine. Morphine, codeine, these plant extracts, but they are used as a alkaloids here. Illegally also used this in different ways here. So that here morphine, codeine, alkaloids, terpene, monoterpene. Terpenoids are monoterpene, diterpene. What purpose using monoterpene, diterpene? They have been used in industry for preparation of different colors. Color industry, varnish industry, terpenoids are used. For preparation of perfumes, lemon oil. This is the lemongrass oil. Essential oil is the lemongrass oil. Lemongrass oil is the essential oil using for preparation of perfumes. Taxines, heparin, resin. Here, heparin taxine, resin taxines, they have been used. In pharmaceutical industry, these taxines are used. Cone cannavalin, lectin, cone cannavalin A, cannavalin A. It is lectin. Drugs, cancer drugs, vinblastine, vincristin, curcumin, here vinblastine, curcumin. These are what is here curcumin, vinblastine drugs. So what are these here? These are the different secondary metabolites. That means the chemicals are biomolecule. They have been produced in the plants. They are helping for indirect growth and development. Those are called as the secondary metabolites. But these secondary metabolites, what are these? They may be pigment, they may be lectin, they may be terpenoid, they may be alkaloid, it may be drug, it may be taxine. Different kinds of these here categories will be there. In drug category, what will come here? Vinblastine, curcumin will come. But these drugs, what purpose using here? Cancer, anti-cancer. For treatment of cancer, vinblastine, curcumin is used. Lectin, concanavaline A. Pharmaceutical industry, these lectins are using. Heparin and resin, what purpose using these taxine? They are also used in pharmaceutical industry. For preparation of various pharmaceutical products, heparin and resin using. Yeah, perfume industry, you see, an essential oil. It is produced in plants, but not for plant welfare of man. What purpose it is using here? Perfumes preparation, lemongrass oil. Terpenoids, what are the terpenoid? Monoterpene, diterpene. In some stems here, in the cortex region, these terpenoids have been stored. These terpenes, they have been collected outside. They have been used in varnish industry, color industry, different paints, Asian paint you are using. In this year, what they are using here, these terpenoids, they can be used. Color industry and different, what would be there? Varnish industry, it can be used. You know that very well, morphine and codeine, these are what would be here, these are alkaloids. But by means of acetylation process of morphine, what has been produced? Heroin. Heroin means brown sugar, illegally using this drug. So here it is also painkiller. Morphine is also. But here it is a slow down the body function. It is helping for what happens here? Rejuvenate the body. When they are at 60, 70 years old, they are looking like young. That means it can rejuvenate the body. And it is painkiller. Who are boxers and sportsmen, then when they are getting used pains to relieve these pains here, like these drugs, they are abusing. But it is addictive nature. Once it has been taken, if without this drug, body cannot do normal function. Therefore, here the secondary metabolite, morphine and codeine, example for alkaloids. In plants, chlorophyll, primary pigment, directly involving in growth and development. But carotenoids, here it is supporting. These secondary means not directly involving metabolism, supporting disease. That means here indirectly involving in growth and development. Carotenoid, indirect growth. Anthocyanin, it is also indirect growth. Therefore, here carotenoids and anthocyanin, they comes under what category? Secondary metabolites. Normally, these secondary metabolites, next gums are also there. Cellulose, polymeric substances, gum, cellulose. In some plants, you see acacia species is there. On the bark of acacia, what will come? Gum. Then now you are preparing Fevikwik here. Suppose any paper has been turned out here to stick this here, gum using. Gum, what is the product? Plant product. So that is secondary metabolites. So here you see drugs, next polymeric substances are there. What is the polymeric substances here? Polymeric substances are gum is example for polymeric substance. Rubber is example for polymeric substance. Cellulose is example for polymeric substance. Gum is there. Gum what purpose using different products? These all man welfare only. Rubber, plastic preparation, different rubbers using in various industries here. Rubber is also product of plant. From latex of plant, Hevia brasiliensis plant here, latex has been taken, industrially processed to make the rubber. This rubber it has been used in various industries, maybe electrical industry, plastic industry,
various industries rubber has been used. Cellulose, what purpose using cellulose? Paper preparation. Paper pulp, what would be there in paper pulp? Cellulose only. So here, this is a polymeric substances here, gum, rubber, cellulose, secondary metabolites. So here, secondary metabolite means various biomolecule, they have been produced in the plants, but involving in growth and development indirectly. But these secondary metabolites, they have been taken outside by man using for man welfare. These are the primary metabolite, secondary metabolite, acid soluble molecule, and acid insoluble biomolecule. Estimation of biomolecule by using trichloroacetic acid method. These three concepts here today we have discussed in introduction point. And next class here, what biomolecules, what are amino acid, what are proteins, what are nucleic acid, what polysaccharides, those significance in plants, and detail all biomolecule, including enzymes, our syllabus it is there. This will be discussed in further class. Okay, thank you one and all.